Um, yeah, I'm finishing up my PhD right now, and I'm based in Oslo at the Nick Walls Institute at a private hospital, and then also affiliated with the PsychGen Center for Genetic Epidemiology and Mental Health, and that's at the Norwegian Institute of Public Health. And so I'm quite interested in neurodevelopment and genetic and environmental effects in the development of that, as well as the sort of overlap between neurodevelopmental conditions and neurodevelopmental traits in the general population. So this is a fun paper to present on because it sort of gets at a lot of that. Um, yeah, should I go ahead and start? Great. Yeah, so this is a project we've been working on, and it's looking at direct and indirect effects of early neurodevelopmental traits. And this study is done in the Norwegian mother and father and child cohort study. And I think I've put together quite a bit to present on. So if there's stuff that I'm taking too long to explain, just go ahead and you're familiar with, tell me to speed up. Likewise, if there's something you'd rather me expand on more, just let me know. And I'm happy to take questions throughout. Yeah, so just a bit more, I guess, of an introduction. I said I'm from the PsychGen Center of Genetic Epidemiology and Mental Health. We're based in Oslo. This is not photos of our center, the photos of just downtown Oslo. We're based over two institutions, Lovisenberg Hospital and then the Institute of Public Health. And we're a collaboratory multidisciplinary team, mainly of geneticists, epidemiology, biostatisticians, psychologists. And we typically look at a lot of different sort of questions of genetic and environmental influences on mental health and various confounding factors. And when I'm saying we, I've borrowed the slide and it's a bit out of date, but I wanted to include it because it has quite a few photos of some of the members of the team to put some faces to when I just vaguely say we. And there's also a few examples of some of the projects that are being worked on in our group. And these blank faces have been hired now, so that's another update. And a lot of the work that we do and what I'll be presenting on today is done in the Norwegian mother and father child cohort study. I'm not sure how familiar you are with it, but it's a Norwegian population cohort, and it was a birth cohort. So pregnant women were recruited between 1999 and 2008, and about 40% of women that were asked were uh, uh, agreed to participate. And so now, when I'm saying children, it may be good to keep in mind that they're now in young adulthood or late adolescence. Although this study that I'm presenting on is primarily using, or it is using their age 3 data. And so it's quite a large study. In the end, there's 414,000 children in the big overall sample. And some of those are siblings, so there's 95,000 mothers included, and then quite a few of the fathers have also included. And the study overall, gave reoccurring questionnaires over time. And so there's lots of data collected on environmental exposures. The earlier questionnaires have lots of questions on neurodevelopment, also mental health, especially in some of the later questionnaires. And the data is also linked to various registries in MOBA, such as like primary or specialty healthcare, but then there's also linkage to school performance and grades as well. And just to visualize this a little bit more, this is sort of an overview of a lot of the questionnaires asked. And from this slide, what I want to sort of point across is that I will just be presenting on a little bit of this data and the data that was collected at the age 3 questionnaire, so 36 months. But there's sort of a wealth of information that can be looked at. And one of the big sort of strengths of MOBA is the trio structure. So we have genetic data on almost 80% of the cohort. And so we have the genetic information of not only the children, but also the mothers and fathers. So that's quite important for when you want to look at contributions of indirect and direct effects. And that's what this paper's focus was on. And so just to define some terms and how I'm using them, direct genetic effects there, that's what I think typically think of when you think of genetic effects, and it's the effects of the individual's phenotype, or the effect of the individual's genotype on their own phenotype. So that's what you would generally consider to be the primary component when you're estimating heritability and such. And then what I'm saying, indirect effects, and sort of the most simple definition would be the effects of another individual's genotype on a different individual's phenotype. And so in the context of a family structure, that might be the mother or father's genotype may influence some sort of environmental factor that then influences their child's behavior. And that may show up in as being heritable in a GWAS or an estimating heritability just using the children because the parents' genomes are correlated with the child's genome. And so that sort of mechanism by which it's acting through an environment that the parents create is what sort of been termed genetic nurture, excuse me. And so this is a figure that I pulled from a paper that outlines or is also a paper in MOBA, and it outlines one of the methods that I'm going to use in this study. But I included it here because I think it's a good diagram to show the different pathways. But here in this sort of lilac blue color would be representing the genetic transmission, where the green is sort of representing the indirect effects or the genetic nurture, if you want to interpret it that way. And then so by where this paper looked at ADHD traits in children. So for example, the mother's polygenic score for ADHD would influence their child's ADHD traits via transmission of the ADHD liability to the child, which then is expressed from the child to their traits, but also potentially via other pathways, such as the environments that they create. I also tend to use indirect genetic effects instead of genetic nurture, and that's just because there may be other effects other than what you would consider genetic nurture that seem to be biasing the indirect effects, such as population stratification and selection biases and things like that. They seem to be getting captured in that estimate. And so, sorry. This is the paper that I'll present on. It's up on medRxiv if you want to take a look. But with that, I just want to sort of do the disclaimer that it hasn't been peer-reviewed yet. So the results are best interpreted as preliminary, and we do see a few odd things, especially in the indirect effects. And so I'll point those out, and there's stuff that still needs to be looked into. So just keep that in mind as I present. And so the aims of the study were to first quantify the effect of direct and indirect effects on early neurodevelopmental traits, sort of the overall magnitude. And then we also wanted to look at how much of indirect and direct effects are being captured in established polygenic liabilities, particularly neurodevelopmental conditions, and then related traits, which we looked at educational attainment and cognitive ability. And we used two different within family approaches to address these questions. We used a variance decomposition method, and that was to look at the overall effects and addressing this first question. And then we also used trio polygenic scores, incorporating the polygenic scores of all three members of the trio in one model. And that was look at the second question. So just a bit more on neurodevelopmental conditions and traits, just in case you aren't familiar with the phenotypes, but there's several sort of definitions of what might be included as a neurodevelopmental condition, but here I'm sort of going with what is defined by the DSM-5 and ICD-11. So that's conditions such as ADHD, autism, which are sort of most talked about, but also intellectual disabilities and specific learning disabilities and other conditions like that. And these conditions are all characterized by significant difficulties in the acquisition or execution of developmental skills. And they typically occur early in development. We're looking at age 3, and that would be sort of autism might be starting, and intellectual disabilities might be starting to be picked up around then, but it's a little bit before the development of most of these conditions. These conditions also have quite high estimated heritabilities. That's from twin studies. Also, the GWASs of ADHD and autism show that there's common variant getting picked up there too. And then the genetic variation associated with neurodevelopmental conditions is shared with neurodevelopmental traits in the general population. And what I mean by neurodevelopmental traits there is just variation in different developmental domains that are generally associated with neurodevelopmental conditions. So for example, genetic liability to ADHD is associated with variation in attention and hyperactivity and impulsivity in the general population. And I want to just expand on that point a little bit more because me and others have looked at just the overlap on genetic liabilities for these questions and traits in MOBA in particular. And so here's some results from a previous study by Ragna, who was a PhD student in our group. And she looked just at the child's ADHD polygenic score and the autism polygenic score and the association with various different traits across a series of the MOBA questionnaires. And so I particularly want to point out that she looked at age 3, which is the same data that I'll be using. And it was on a smaller subset of the cohort because not everybody had been genotyped at that point. But specifically, what's important to point out here is that she did find associations with the child's ADHD polygenic score. And let's see, can you see my pointer if it seems to be freaking out, but it's associated with hyperactivity and inattention at age 3, and then the autism polygenic score was associated with increased motor difficulties at age 3. And so, there we go. Next, so if we know that the child's polygenic score is associated with some of these traits early, now we sort of wanted to look at potential indirect contributions. And from my knowledge, when looking at neurodevelopmental traits in the general population, most studies have focused on inattention, hyperactivity traits, and ADHD. And so at older ages, in MOBA at age 8, variance decomposition methods, very similar to the ones that I'm going to use or present on, found both indirect and direct effects underlining these traits, but the direct effects were of, I believe, twice the magnitude in both of those papers to indirect effects. Polygenic score methods, both in TEDS and I think this paper, if I remember correctly, in MOBA, and this paper, I believe, was in TEDS, find primarily direct effects underlying ADHD traits at age, I believe, is like 8 in MOBA and 12 potentially in that other study. So what we wanted to do with this paper was look at a broader range of traits and also at an earlier age. And so these are the areas of development we looked at. We looked at social communication, repetitive and restrictive behaviors and interests, difficulties in attention, hyperactivity and impulsive behaviors, motor development and language development. We used the social communication questionnaire, which is an autism screening questionnaire to measure social communication and repetitive and restrictive behaviors. We used the CBCL for attention and hyperactivity domains. And then we used the motor and communication subscales of the ASQ for motor development and language development. And so our sample size—
Just a quick question. Sorry to interrupt. This is probably obvious, but just want to clarify. These are questionnaires targeting parents, right?
Yes. Oh, yes, yes. Very crucial point. These are all maternally reported.
I see. So
Yeah.
when people study directly, in fact, maybe this is less of an issue, but if you actually want to understand how parents' genome influence children's phenotype, whether the quality of these answers they provide in these surveys, do you think that could be a factor that influences your results?
Yes, that's exactly right, and a bit of a spoiler, but we'll get to
Oh,
that.
yeah, I'm happy to wait that. Yeah.
No. Yeah, I think one thing we really need to consider is, yeah, what these measures are capturing, especially at an early age. For example, the social communication questionnaire was designed for like ages 4 and up. So it's a bit early to, and it has sort of mixed sort of psychometric properties for before that. So yeah, what is being captured in the questionnaires is definitely something to consider. Then also the fact that they're maternally reported and how that might be impacting, and the fact that they're asked in a general population as part of a large survey. So it's not, the parents may not be familiar, for example, with what the questionnaires are actually getting at or the behaviors they're trying to measure. So that's all things that I think, yeah, should be considered.
Thank you.
Yeah.
I
Can I
had a
ask a, oh, sorry,
yeah.
go ahead.
I had a really quick question. Just when you say unrelated trios, is that adoptees or?
Oh, sorry. So no, so there's some quite complex family like relationships in MOBA. So we calculated the genomic relatedness matrix. So it's like removing, for example, half siblings, cousins, that sort of thing. So you might have a trio where the parents have a sibling who's also a participant in MOBA, and so the kids are cousins, and things like that. So we use the complete sort of all the trios in the polygenic score analyses, and did some sensitivity analyses, dropping like siblings and half siblings, and used sort of cluster standard errors. But with the trio GCTA method, we needed to restrict to completely unrelated trios, and so there we did that based on a genomic relatedness matrix and filtered so that I think on 0.1, so cousins and above would be removed. Yeah. So yes, that's the sample sizes.
I have a quick question as well about
Mhm.
the questionnaires. Did
Yeah.
you also, or did MOBA also get these questionnaires to be filled out by the fathers?
So the fathers answered questionnaires at 2 points. Once I believe was around birth, and then once was later, but we do not have it on age 3. And
Okay.
then I think the father's questionnaires were primarily on themselves and not reporting the children's behaviors. So yeah. Okay. And then for polygenic scores, which we picked, we wanted to focus on sort of well-powered neurodevelopmental condition scores. So we used ADHD and autism. We also included dyslexia, the recent one, although there we only had the top 10,000 publicly available SNPs. So that score is, you'll see it's not picking up much. And then for associated traits, we looked at educational attainment, and then I'll be saying cognitive ability, but it is the 2018 sort of intelligence GWAS. And we picked educational attainment and cognitive ability because they are sort of associated traits to neurodevelopmental conditions. Educational attainment is positively or genetically correlated with autism, and then negatively genetically correlated with ADHD and dyslexia. Then it's also from PDTT analyses. We know that educational attainment is overtransmitted in autism cases and cognitive ability undertransmitted in ADHD. And so that, if you're not familiar with the PDTT, I have trouble pronouncing that method. That's just looking at, for example, in trios where the child has autism and the parents do not. It's looking at if autism polygenic liability is over transmitted to the child who has autism based on the mid-parent score. And so that what that means is just that that's sort of controlling for a lot of population stratification, and is more robust to different selection biases. So that association seems to be, a direct sort of genetic effect and not completely sort of indirect or based on that sort of thing. And then there are also indirect, our educational attainment and cognitive ability are interesting traits to look at in the context of indirect effects, because from the within family GWASs, we know that these are traits that seem to have substantial indirect effects that are biasing the heritability estimates when you look at the GWASs in a population-based sort of GWAS. So that was the rationale behind choosing these scores. Yeah. And so I've mentioned them both now, but we use two different methods to address our questions. And one of those is called Trio GCTA, and it's variance decomposition extension of the GCTA methods. And it's used to estimate, and we used it to estimate the sort of latent direct and indirect genetic effects that could be attributable to the measured SMP or common SNPs in MOBA. And then we also used Trio polygenic score method, and there that was to investigate the direct and indirect effects attributable to specific polygenic scores that I just mentioned. So to explain the method a little bit more, so for Trio GCTA, as I mentioned, it's an extension of the GCTA. And so there, instead of just estimating the additive genetic effects of unrelated individuals on a trait, we're expanding it to include mothers, fathers, and children. And so this method was developed by Espen, who is the second author on this paper. This paper right here outlines the method if you're interested. And he also has the code and a package that he's developed to run this up on GitHub, if that's something you're interested in. But I will try to explain it briefly. So a sort of simple, the theory behind it can be sort of expressed here in a simple sort of linear model of the phenotype, where we have Y, and in our case is the early neurodevelopmental traits. And instead of just estimating the random effects of the child's, or the variance due to the random genetic effects, or additive genetic effects, excuse me, of the child's genome, we've also then included parameters for the mother's genome and the father's genome. And so the covariates are still included as fixed effects, and then the additive genetic effects are estimated as random effects in the model. And then, of course, they have the residual effects. And we do this by estimating the genomic relatedness matrices for the entire sample, and then split it by mothers, fathers, and children. And so if you have sort of trio number 1, that member of the trio will always be in the first row of that matrix. So for example, in trio number 1, the child will be the first row in the genomic related matrices for the child. The father will be in the first row for the genomic relationship matrix of the fathers, and then so on for the mothers. And that's sort of the basic model. I hope that's clear. And then sort of looking at the variance decomposition. So that can be decomposed if we have the variance, in our case of the early neurodevelopmental traits, we're estimating then the variance attributed to the mother's phenotype, or the mother's genotypes, the father's genotype, the offspring's genotype, and then also contributing is the covariance between the child and mother's genetic effects and the child and father's genetic effects, and then of course the residual variance. You may notice that the covariance for the between the mother and the fathers is not included. And that's because we're making the assumption that there's no sort of mating's random. So then we've constrained that to 0. So that's not contributing to the variance. Also, what we are interpreting as direct and indirect effects depends on your outcome. So for us, it's a child's outcome, and we consider the child like the focal individual. So there we will consider the child's or the variance attributable to the child's genetic effects as the direct effects, and the variance attributable to the parent's genetic effects as indirect effects. But if you were to say, look at maternal depression, which was another study that was done in MOBA, then the effects due to the maternal genetic effects would be direct effects, while the parents and offspring would be indirect effects. When we're looking at child's phenotypes, you can also, or the covariances here are capturing or quantifying how much the same variants contribute to direct and indirect effects. In sort of the context of the family trio, if you're interpreting the parents' indirect effects as contributing to the environment that they raise the child in, then that could also be interpreted as the variability in the phenotype due to gene-environment correlation. So positive covariances between the indirect and direct effects would increase variability or variance explained in the phenotype, and negative would decrease variance explained in the phenotype because they would sort of counteract each other.
Laura, can you say any more about this assumption about random mating? Like, what if you didn't make that assumption, does everything fall apart? Or I guess I wonder how important this is for the model.
Yeah, this is a very good question for Espen. But we did that because, I mean, we looked at correlations, and on average, the parents' genomes aren't correlated. So yeah, it seems fine. But I think there's more evidence that a sort of mating may be contributing to traits, and specifically potentially when we're looking at things like educational attainment, if those are influencing any of our outcomes, that could be something to consider. But to be honest, it's an assumption we make, and we haven't looked too much into how that might be affecting the model and what could go wrong if it's violated. So that's something to look into for sure. Yeah, sorry, I can't expand further on that. Yeah, and so what I just explained was the full model. We also compared fits between simpler models to the full model, just to see if the effects were better explained, not including some of these parameters. So we tested the full model, then we also tested a model dropping the covariance terms, and then a model dropping the indirect effects, and then a null model. And so we used the AIC to sort of select our model, but we also looked at sort of formal fit or formal comparison and the BIC. And so in the paper, we've ended up presenting all of the results because there's a bit of sort of bit more of an art, I guess, in selecting the best fitting model. So keep that in mind. And so to get into some results, for 3 of the outcomes, based on the ASC and marked here with a star, the best fitting model was this no covariance model. So that's this one right here. And then the percent of the variance is on the y-axis, and the maternal effects are marked in purple. The child's effects or the direct effects are marked in blue, and then paternal in this sort of light green. And then the covariances are here in the yellow and the darker green. But we primarily then are interpreting the direct and indirect effects because all of the models were significantly, it didn't lead to a significant decrease in fit to drop the covariance parameters. So for intention, we see that there's sort of equal direct and indirect effects estimated, but quite unexpected. We see more maternal indirect effects estimated for the hyperactivity and repetitive restrictive behaviors. In fact, very little direct effects estimated, with standard errors greater than the actual parameter estimate. And that was a bit unexpected. We didn't try models that only included indirect effects. So that may be something to look into. But yeah, so it seems to be primarily maternal indirect effects for those two phenotypes, at least based on the trio GCTA model.
I wonder if you can comment on the negative correlation of direct and indirect effect when you do have those terms in the model. It seems to be a fairly commonly reported observation in the literature, but personally, I've never understood why there should be a negative correlation.
Yeah. So it may be just what in overall the variances explained are very small. None of, for example, the indirect effects, direct effects, none of those are over, 10%. Some are as low as 5. So then the variance explained by the correlations, you wouldn't expect that to be much larger, I think.
Right, but I guess I'm not claiming these are statistically significant or anything, but if you look at the panel on the right-hand side, the yellow box is bigger than child direct effect, actually, right?
Yeah. I guess that it means then there's a strong opposite effect overlap between what is included in the direct effects and indirect effects. Actually, that's something that we should look into. We haven't, to be honest, looked tremendously into these because it was fine to drop them. So
yeah, I perfectly understand because you said the model without these covariance terms gave you better model fit, right?
Yeah.
But it's something that has puzzled me.
No, it's
in the past
puzzling me
because
too right now.
when you estimate these parameters, they seem to be negatively correlated. It's like the child genetics that increase some outcome would decrease outcome if it's the effect from the mother, for example. Just it doesn't make intuitive sense, but it's perfectly okay if you have it dig into it.
No, I mean I think it's basically that whatever effects are in the child are not the same genetic effects. I guess in this case it's mainly the father that are decreasing, so they're acting in opposite directions and canceling each other out and decreasing the variance. Putting like an actual example to what that might be, that's where I think it gets tricky and a bit speculative. So yeah.
Thank you.
Yeah, and then for 3 of the phenotypes, the model that was the best fit was just direct effects only, so no indirect effects. And there we sort of see about the same, about 5% of the variance could be explained by the child's genome. And so that was for language, motor, and social communication. And so now moving on to the trio.
Laura, I have one question about the different phenotypes. Have you gone back to look at the items in these different scales and whether these 3 seem to be more quote unquote objective, or like what maybe a clinician would say, as opposed to the other 3 where maybe the mom would? So yeah, could you have any comment on that?
Yeah, I think that's so. Yeah, I think that's definitely potentially contributing. The ADH, the ones covering attention, hyperactivity, and repetitive behaviors are quite well endorsed in the sample. Especially, some of the repetitive behaviors capture speeches. I believe like one of the questions, for example, is does your 3-year-old ask socially inappropriate questions? I think it may be more say yes to that than no. So there are things that are being more frequently endorsed in the sample. the measures of language, motor, and social communication are much more rarely endorsed in the sample. There are going to be some questions, for example, in the language development that would be getting at not having functional speech yet at age 3. So they are, I think they are probably picking up on more clinical phenotypes than potentially the other measures.
Is there a way to know, because these kids get older in the data set, can you do anything more formal about these different domains as it relates to their future clinical or their future diagnoses or anything like that in the sample, or am I misunderstanding what's possible with the data?
Yeah, well, we can see, for example, who's received diagnoses of autism and ADHD. I've had another paper that has looked at a lot of these domains at age 3 and see how they relate to later diagnoses. And it does seem that like all the domains by themselves are associated with receiving diagnoses of pretty much every neurodevelopmental condition. When you're controlling for the effects of all these domains, the social communication and language and motor actually are stronger predictors of receiving, for example, an autism diagnosis than the repetitive behaviors. Potentially that's again what the measures are capturing, or maybe that's a phenotypic difference, but yeah. I do think potentially what we're seeing is these measures are capturing, yeah, a more clinical phenotype. Or a less subjective phenotype, potentially. Yeah, so now the trio polygenic score method. So here we're using these continuous neurodevelopmental trait measures, the one that were used in the trio GCT models, as outcomes in just a multiple regression model where we've included the polygenic scores of all 3 members of the trio, along with covariates. So with 6 phenotypes and 5 polygenic scores, that ends up being 30 separate models. Yeah, we also had originally wanted to look at sort of subdomains of motor and language, that being gross and fine motor, and then receptive and expressive language. There with the distributions, it wasn't, because some of these phenotypes are so rare in the general population, we didn't look at them as continuous measures and instead ran some models with like logistic or dichotomizing the variables, looking at like ever reporting difficulties versus not, and looked at them in logistic and ordinal regression models. And then also as a bit of a sort of somewhat analogous method to the trio GCTA, we wanted to look at the variance explained by the scores that we looked at in the phenotype. And so there we looked at partial R-square values, splitting where we ran a model with all of the polygenic scores included in one, and we didn't interpret the beta estimates there since they lots of multicollinearity because some of these like including educational attainment and cognitive ability in the same model. So there we just looked at the partial R-squares, splitting the effects into direct based on the child's score and then indirect based on the mother's and father scores. And so there we were more looking at like the ratio between direct and indirect effects and not necessarily the actual values, since the variance explained by polygenic scores is so small. Yeah. So getting into results from these, first, just looking at ADHD and autism. There were for ADHD, the only direct effect we identify after multiple testing corrections is an effect on attention. There was an effect on hyperactivity, however, after multiple testing corrections, that goes away. And then for autism, we see it is associated with language difficulties. And then sort of oddly, we see a negative association with attention. So that would be increased autism. Genetic liability was associated with lower reported attentional difficulties. We also see a few indirect effects popping up. They're primarily, or they all are in the fathers. Some, such as ADHD and attention, are in a direction that you might expect, but others, not so much. For example, this autism, polygenic liability being associated with decreased language difficulties in children. Yeah, I'll talk to a bit more about that. Unclear exactly what's going on, if that's representing actual effects or potentially we are selecting on complete trios, and fathers are typically the member of the trio that is missing. So that's something to consider. And then looking at the subdomains, we see that the those indirect effects do still show up in the expressive language for the autism score in the fathers, and then specifically fine motor skills with the ADHD score. The direct effects of autism on language didn't survive multiple testing corrections, but I think specifically with this receptive language, those traits were quite rare in the general population, so we don't have a tremendous amount of power there, so it's quite an imprecise estimate. And then again, I'd mentioned we'd also looked at the dyslexia score. That was only the top 10,000 SNPs, so we don't see much. And this is also an early age where learning disabilities are typically diagnosed more once children start school. We do see an indirect effect pop up with language in a direction that you might expect. And then when we looked at the subdomains that remained in the expressive language, and a direct effect actually on motor popped up just for gross motor, where increased liability to dyslexia was associated or decreased the odds of ever reporting gross motor difficulties in the children. And so now looking at the cognitive ability score and the educational attainment score, here we see a lot of things popped up. These are also, quite well powered in comparison to the neurodevelopmental trait polygenic scores, so that should be considered. For direct effects, we do see that after controlling for the parents' genetic liability to these traits, that there are still direct effects in the directions that you would expect for attention, hyperactivity, and language, for cognitive ability, and then EA for attention. And again, since these are quite correlated, a lot of these effects, when you see them both, or one that's significant and one wasn't significant anymore after multiple testing correction, they're sort of capturing probably a lot of the same thing. We also see quite a few indirect effects. Some of them are in directions that you'd expect, such as decreased EA liability in the fathers to lower, or actually no, that one's in the opposite effect. And then I also want to highlight, for example, this maternal indirect effect here, because this one's in the opposite direction than you expect, where higher liability to EA is associated with more maternally reported hyperactivity traits. So that's a bit odd. And then we see some effects, indirect effects are found in both of the parents. So for example, in motor, we see that higher EA and cognitive ability are associated with reporting more difficulties in motor development. And what's interesting there is that when we look at the subdomains of motor, this seems to be in the fine motor. And then there is a direct effect identified in the gross motor that wasn't identified overall. And then we see the indirect effects stay with EA for expressive language, and the direct effect is there for the cognitive ability score in expressive language. So yeah, that's quite a lot of results all at once. Is there any questions on that?
Just to take the fine motor as an example,
Yeah.
Laura, is there how, if you go back to this, like the items that the mother is asked, would you describe them as quantitative, or that there's an implicit sense that or maybe that the reporter thinks, as compared to other children my child's age or something like that, that it's not actually saying something very specific, but there might be some this might be produced by anxiety of parents or something that's not being could you comment on that?
Yeah, so they ask for specific skills, but for example, the fine motors is, I believe the two questions is like, or two of the questions are like, does your child have trouble buttoning and not buttoning shirts? And then another one is about how they hold a pencil. For gross motor, I can't remember all of them, but I know one is like having difficulty kicking a ball or standing. I do know that the fine motor questions, again, are much more well endorsed in the sample than the gross motor. The gross motor is quite infrequent for parents to report, especially the way the response style was for these questions. It was trichotomous, so it was like never, sometimes, always. So it's quite rare for the parents to reporting some of the gross motor difficulties like always and all the time. And so that's where, again, it seems to be getting at a rare sort of phenotype where potentially the fine motor has more subjectiveness. Yeah.
I'd be interested in something on the order of asking the parental, especially the maternal PGS for like anxiety or something, relate and see if any of those pop up in a similar way
Yeah.
for just a strictly reporting question, really. Then
No, I'd be interested in that too. I know the study I referenced earlier on the ADHD traits with the trio polygenic scores, there they found the one indirect effect that they found the most robust evidence for was neuroticism on higher reported traits for, I think it was, yeah, attention. I think it was hyper. I can't remember if it was a sum score of all the attention hyperactivity or if it was just hyperactivity, but yes, I think it would be quite interesting to see, yeah, anxiety as well, and other traits that could be getting at what might influence a parent to report more or less. Yeah. And then I can speed through this a bit if you want, but I just wanted to sort of show the results of the two methods next to each other. But for the trio GCTA, for attention difficulties, we find sort of small but equalish amounts of direct and maternal indirect effects. Based on the trio polygenic score analyses, there we see that some of these direct effects can be attributable to genetic liability that's been associated with ADHD, cognitive ability, and EA. And potentially that autism score came up. We also find paternal indirect effects when using the polygenic scores. That's something that in the trio GCTA model, that parameter was estimated at 0. And that I just want to use as an example. We see some different results between, finding paternal indirect effects in the polygenic scores, but not in the GCTA. On there, I think one of the main things to consider is sort of the scale at which the polygenic scores are operating at in terms of the variance explained, because it's, I mean, 0.13% in the sort of larger scale of the genome wide, and just comparing model fits, that's not going to come up. Then I also think, especially with things like EA, you have to consider effects that are getting captured in those population-based GWASs, particularly, yeah, sort of like selection or indirect effects there. So that's one thing there to note with differences. For hyperactivity, impulsivity, we see primarily maternal indirect effects. There, based on the polygenic scores, we see higher EA and cognitive ability is associated with more hyperactivity and impulsivity symptoms or traits. And then we also find some direct effects in the opposite direction for those scores. For repetitive behaviors and interest, again, the most evidence for maternal indirect effects from the trio GCTA models, and the polygenic scores show that potentially some of that's being captured in cognitive ability. Then for language, we see primarily direct effects in the trio GCTA models that, according to polygenic scores, may be captured in autism and cognitive ability. And then we also identify some of those paternal indirect effects in the trio polygenic score models. For the motor domain, again, primarily direct effects. Here we find the strongest evidence for indirect effects actually with the polygenic scores. These dyslexia and EA were associated when we looked at gross motor scores only. There, I think the lack of direct effects found in the polygenic scores, that's sort of also based on which scores we included. You know, we don't have like a dyspraxia, for example, polygenic score. So that may be why we're not identifying direct effects. And then lastly, like social communication, there are some small direct effects were identified in the trio GCTA model. And we didn't identify any in the polygenic scores, and just some evidence there for the paternal indirect effect from the ADHD score. And so with that, I just want to talk a bit more on the indirect effects. I'm hesitant to interpret them all as genetic nurture, at least at this point. What is on this slide is all speculation, of course, but just to offer some potential other things that I would want to sort of look into, is sort of the effects of selection and biases in MOBA. You know, we have sort of three layers of selection, first into the cohort and then staying into the cohort by age 3, having genetic data, and then also being a complete trio with genetic data. We know, for example, both the language and the social communication had some of those paternal indirect effects come up, and we do know that both of those scores, that incomplete trios, had lower or significantly lower reports of social communication, or significantly higher increased reports of social communication difficulties and language difficulties than the complete trios. We also know that, for example, parents with children with neurodevelopmental conditions are dropping out of MOBA at higher rates than children or parents without children with neurodevelopmental conditions. I'm also curious about, especially with the maternal indirect effects, and when we identify maternal indirect effects and no paternal indirect effects, how much reporting biases may be playing into that finding. Then there's also sort of, you know, called dynastic effects, especially with associations with the EA and cognitive ability scores. That's basically sort of SES getting captured in the genetic nurture. So sort of multi-generational transmissions of sort of social capital and wealth is getting associated with sort of residual population stratification and getting captured in that effect. And then also sort of not just considering what's in MOBA, but for the polygenic scores, what might be getting captured in the population-based GWASs that we're using to construct these scores. So yeah, that's sort of my thoughts on what I would want to look into more before assuming genetic nurture effects. But yeah, all sort of speculative there. And so just to sort of wrap up, our conclusions here are that there's various pathways of genetic transmission that seem to be involved in reports of early neurodevelopmental traits in the general population, and that seems to differ on the sort of contributions and where it may be coming from depending on specific areas of development. So indirect effects may be more something to consider, for example, early reports of the ADHD related traits or repetitive behaviors than for the gross motor or such. And then I think more broad than just neurodevelopmental traits, I think using complementary methods, such as the variance decomposition and the polygenic scores, sort of give insight into these processes at both, quote, like a macro and micro level in terms of getting an estimate of the overall magnitude from the variance decomposition, but then being able to say a little bit about some of the mechanisms behind these pathways using the polygenic scores. And yeah, I think it sort of highlights the strength of using within family approaches when possible to be able to disentangle some of these processes and what might be some of the genetic contributions to the phenotypes of your interest. Yeah, and just some acknowledgements. Obviously, this was work from a big part of a team. I think if you have specific questions, I can direct you that I can answer. I can always direct you to people who are more in a better position to answer that. And yeah.
Thank you. Let's line up for questions. Yes, and thank Laura for the great presentation. I guess we could. So maybe we could try if you could raise your hand just to get you in order that we could get the questions going. How about that? So let's start with Q.
Yes. I wonder if for a subset of children in the mobile cohort, you actually have access to clinician diagnosis, or do we have a sense of just what's the fraction of kids that actually received ADHD diagnosis, for example?
Yeah, so we have linkage to specialized healthcare, and so it's registry data. So we have like, has this child received a diagnosis code? For, I believe, a subset of the cohort is also in the autism birth cohort study, and there's more detailed information. I am not tremendously familiar on exactly the overlap, but I know that's available. So I have looked at diagnoses with the kids in age 3. It's a bit hard to tell with the registry data when exactly they got the diagnosis, because the registry data starts in 2008. And so we know that most kids who had gotten their diagnosis before that will get it later on. So they're still marked as having a diagnosis, but whether or not that had occurred yet at age 3, we're not entirely sure. I mean, that's most relevant for autism and general developmental delay and things like that. But yeah, so off the top of my head, I think, I know overall in MOBA, there's around 1,800 kids with autism. And I think, oh gosh, I'm hesitant to say for ADHD because I don't want to be wildly off, but I think it's around 3,000. That'd be something I need to check. With age 3 data, that obviously decreases. Yeah,
No, that's okay. And I understand there's probably still a lot of limitation in the diagnosis from medical record, but I wonder if you can comment on the pros and cons of using maternally reported behavioral outcomes versus these medical record derived outcomes in this research.
Yeah, I mean, I think the biggest pro and why we did this is that, at least for a birth cohort, we're going to have very few out of the total sample with these diagnoses. So you can capitalizing on having that large sample size and a quantitative measure of these traits for everybody with the data, with the cohort, instead of just selecting your sample down to your sample of like 40,000 kids down to 1,800. Another for, just purely sort of methodological, the trio GCTA and such, we need a quantitative trait or a continuous measure. So wouldn't be able to use that looking just at, ADHD diagnosis versus no diagnosis. You could look at that with the polygenic scores and such and like logistic regression, but yeah. I think there's also then sort of questions that you can answer. You're getting more at the how much of do these reported traits in a general population sort of correspond to liability that's associated with a condition. And so that's sort of just a separate question that you can look at why using a general population sample rather than just being able to look at genetic effects associated with the condition itself. Does that answer your question?
Yes, it does. Thank you.
Yeah.
Go ahead, Tim. Go ahead.
Yeah, my question is also trying to think about this issue of like, how can you try to validate this measure? So two kinds of issues come to mind regarding why you might have greater indirect effects at this age, right? Like if first of all, the mom is reporting, but also children are probably more influenced at this age than a later age. So you developmentally, you would expect that, right? Indirect effects to be larger at this age. That probably was the reason why you were studying.
Yeah, and I think it differs from the older age, and I think that that could be a reason. It's something that we'd probably want to look at instead of just speculating, like extending potentially the models longitudinally and confirming that way. But I think it would be interesting to look at some of the twin literature that has looked at this over ages and see what they find there. And if that's
I had two suggestions. So one, if you don't have independent data on diagnoses, but one way one might go about this is, since you mentioned that there are repeated measures, you can look at, you can use state trait models.
okay.
If you're familiar with that. So they're essentially just like trying to find a latent factor for people with repeated measures over time. And you can treat the latent factor as essentially the truth of that person, that true state of that person, the phenotype of that person. Another thing that came to mind is there might be mothers who are, I don't know how many in your sample, such cases are in your sample, but there might be biologically unrelated mothers. And if you could just look at those, it might at least get some sense of how much of these results are being shaped by.
Unfortunately, I don't think we would have biologically unrelated mothers just because they were recruited during pregnancy. But yeah.
Oh yeah.
It would be really cool if we could.
There's that part. That's a good point. But doing this trait thing, that might be interesting. And then using the state factor essentially as the phenotype, the trait factor as the phenotype. I'll stop there though. This is really good. Thank you for sharing.
Thank you. I've written that down. So
James.
Nice job. That was really interesting stuff. And I do have a question and then a comment. So I guess my first question is, a lot of the quantitative traits that you're looking at on the psychopathology domains, they're not going to be normally distributed, right? Some of them you're going to expect a pretty strong zero inflation for, and then others, like you even mentioned for, I think it was hyperactivity, you saw a lot more variation within the 3-year-old sample for obvious reasons. There are 3. So I guess my question about that is, in your analysis, you looked at all of these outcomes in a linear regression model, is that right? How did you account for, please correct me if I'm wrong, because I don't think
for the main domains we did. And that was, we originally, and we had pre-registered this study, we didn't want to look at just overall sort of motor and development. We wanted to look at like the specific domains, and there we were just like, this is not, we can't pretend that this is, continuously distributed. So there we did the polygenic scores and we switched to logistic regression and such. There's still obvious sort of skew in especially like the overall scores for language, motor, and social communication. Again, those are the ones that we're seeing direct effects. They're better and they're more continuous than the others, and we did a transformation to make them more or less continuous, but yeah, that may be
Right,
affected.
yeah, so you're doing
Yeah,
you're
so
doing
it's,
what everyone
yeah.
has to do in this field, which
Yeah.
is to manipulate your data to make it as workable as possible within the context of your analysis. And I guess this is kind of like what I'm hearing in the Zoom call is that we have a lot of questions around measurement. But I want to push back on some of the questions that were maybe or some of the things that were implied here, which is that somehow a medical record is better than what you're doing. I want to emphasize to the group that like medical records, especially for psychopathology domains, is not necessarily better than what researchers can do in terms of actually quantifying through multiple measures on multiple informants of outcomes that are actually not necessarily normally distributed, but are quite heterogeneous in the population. And when we use and worship, you know, whether or not somebody got a diagnosis for ADHD from a pediatrician over a 15-minute interview, I'm not sure that that's actually more valid necessarily than using CBCL, Vanderbilt scores, whatever the case may be. So I actually like the justification that you provided with respect to why there's value in looking at these outcomes using questionnaires, which are flawed, no doubt. I wanted to comment about that too, though, is that because of your focus on 3-year-olds, so a lot of the questionnaires, especially the CBCL, and I'm guessing you're using the MOBA would be using the 1 to 3.5 or 4.5 form, right? Age 1 to 4.5 or something like that. So if you're interested in something like ADHD, that's much like autism, context specific. And you would never diagnose ADHD in a 3-year-old because a 3-year-old is not in enough context for which to express impairment. Like one of the criteria of ADHD is that the impairment across symptoms is seen across multiple settings. And like I said, 3-year-olds are not typically in multiple settings enough for which there's another informant to quote unquote validate that those symptoms were there as well. So I guess I'm more interested in knowing whether or not these predictions, direct and indirect effects, show up in later in the same measures, but at like age 7 or 8 or 9, right? When we have a little bit more confidence about what we're seeing, especially in the ADHD and perhaps even in the language domains. So I think you said that was something on the horizon, that's what you're planning on doing next, or is that not?
Actually, I know, for example, Espen has looked at. He looked at he used age 8 data, and it was externalizing behaviors more broadly, but included the CBCL ADHD measures at age 8. And there he found direct and indirect effects as well, using the same trio GCTA method in this paper. But there, the direct effects were about twice the size of the indirect effects.
That's interesting.
Yeah.
Cool.
Yes.
Yeah, that was great. Thank you, Laura. I guess I just had a question. I mean, also to James's point about the medical records and so I had a question about socioeconomic status and how you view that and whether you know, it's clearly probably an important confounder.
Yeah.
Were you able to look at any of this by socioeconomic status? I don't know if you have the power, but it just strikes me as being interesting.
Yeah. I think that could be an interesting follow-up analysis. We do know that, like looking at broad sort of selection things, that MOBA participants in general, like a lot of cohorts, are more highly educated than the Norwegian population of higher socioeconomic status. We have information on parents' education levels and income in sort of very broad bins at, I believe it's when they first filled out the questionnaire. So that's another sort of how much things changed. But it wasn't something we looked at in this analysis. But yeah, I think it could be interesting to either include that as a covariate or look at interaction effects. That's something that I'd be interested in, but yeah, it wasn't part of this analysis.
Thank you, Laura. Anything else from the group? This has been wonderful presentation, super interesting topic. Thank you for outlining both the data and all the different methods you're using. So I think if there's no other questions, we'll leave it at that. And thank you very much for joining us virtually in our meeting today.
Great, thank you. Feel free to reach out if you have further
Okay,
questions.
bye, Laura.
Thanks, Laura.